It's my great honor to present our research today, minimally invasive sampling for molecular profiling of lymph nodes using a modified acupuncture needle. Currently, blood liquid biopsies have low sensitivity. According to the Revised Starling Principle, tumor biomarker-laden interstitial fluid accumulates in lymphatics. Head and neck tumors often present lymphadenopathy, enabling lymphatic sampling. Yet simple minimally invasive lymph biopsy tools remain scarce, blocking clinical translation. We developed surface functionalized acupuncture needles for minimally invasive lymph node DNA collection with promising translational prospects. First, we fabricated grooved acupuncture needles, coated them with polydopamine, and then modified the surface with DNA-binding histone to get our functionalized needle. We used this needle to collect lymph node DNA for predicting tumor metastatic risk. To boost DNA capture capacity, we etch spiral grooves on needles, followed by sequential polydopamine and histone coating. Grooves strengthen coating adhesion, while histone specifically binds dsDNA. AFM showed a 300 nanometers surface relief after modification, and ES revealed vanished metal peaks plus new nitrogen signals, confirming successful surface functionalization. We simulated tissue barriers for capture tests. Mutton slices acted as barriers, and qPCR quantified GAPDH after incubation. Figures BC show our needle captured 29-fold more nucleic acids than bare needles and polydopamine-only probes, with broad-spectrum binding to dsDNA fragments. Figures DH proved the stable composite coating captures dsDNA efficiently within 5 minutes. We established subcutaneous tumor xenografts in nude mice for in vivo validation. Figure A. Figures B and C demonstrate our needle outperforms bare needles in nucleic acid enrichment and captures more tumor DNA than blood cfDNA. We also built a footpad lymph metastasis model for early diagnosis evaluation. Figure D. Figures F show lymph node DNA achieves higher detection efficiency than tumor tissue and blood cfDNA. No tumor cell infiltration was observed in lymph node H Yu staining. Figure G, meaning tumor DNA accumulates in lymph nodes via interstitial before visible metastatic lesions form. We harvested 15 lymph node samples from 10 patients with head and neck cancer. Whole genome sequencing characterized 14 driver gene mutations. Our modified needle achieved a high mutation detection rate of 86.67%, compared with only 40% for traditional AUE staining. Further sensitivity improvement can be realized via deep sequencing and copy number variation analysis, while targeted sequencing and qPCR can lower clinical testing costs. In figures AB, we compared the detection performance of lymph node DNA captured by our needle and peripheral blood cfDNA. Tests on 4 clinical samples proved lymph node DNA has higher abundance, better integrity and superior mutation detection efficiency. In figures CD, 5 plasma samples and 1 blank control were tested. Only 1 healthy sample carried a variant of uncertain significance in KMTRD, indicating low background noise of our system. Next is the biocompatibility assessment. In figures AC, compared with commercial 5 ml biopsy syringe needles, our needle creates tiny puncture wounds without bleeding. HUE and IHC staining revealed minimal tissue trauma and little neutrophil infiltration. Tunnel's figure D showed far fewer apoptotic cells at puncture sites versus traditional needles. We established a lymph node sampling platform based on functionalized acupuncture needles, applicable to all diseases with lymphadenopathy. It efficiently enriches lymph node nucleic acids and delivers better diagnostic performance than blood cfDNA and HUE staining. We used whole genome sequencing in this study. Targeted sequencing and digital PCR can accelerate detection and lower costs in follow-up research. This platform supports multi-omics analysis, broadening its applications in clinical tumor molecular diagnosis.